very happy to present today on the discovery of SARS-CoV-2 PLpro inhibitors with in vivo efficacy in a mouse model of infection. Um, I'll start with a little bit of background. I'm sure this is familiar to many of you, but just kind of want to put into context PLpro as a target. So prior to our efforts, the SARS-CoV-2 inhibitor efforts were largely focused on the main protease and also the RNA-dependent polymerase. So those, for example, have been shown by nirmatrelvir, which is a component of Paxlovid, and then also remdesivir being approved as a RDRP inhibitor. So if we look at the kind of diagram on the left here that shows the normal viral cycle, you can see that essentially what an MPRO inhibitor is doing is it's coming in and blocking the main protease's ability to cut up the viral polyprotein. This is basically allows you to block the formation of the replication complex, and thus you kill the virus. But there is a second protein involved there, and that is PLpro, and so that is also involved in cutting up the viral polyprotein. Now, there was a lot less experience with PLpro. For example, main protease had been around since SARS-CoV-1 and had already shown efficacy, and also was highly conserved across viruses such as rhinovirus and others, which have kind of a 3CL-Pro. So there was a lot of experience with blocking the main protease. But PLpro is kind of a different story. There was pretty limited data that it could lead to really potent antiviral effects. But it is, again, cutting the viral protein at 3 sites as opposed to the 11 of the main protease. It also cuts itself out, which is quite interesting. I'm not going to go into that biology, but there is a lot of interesting biology there. In addition, it has this kind of immunomodulatory effect. So it's not only processing the polyprotein, it is also combating host immune response through a kind of deubiquitinating and de-ISG-lating activity. It's super interesting, this immunomodulatory effect. I'm not going to go into it much today, but hopefully some of the compounds we produce can help study that functionality further. So I'll first mention this project was done in collaboration with Pfizer. So my co-first author on this paper was Michelle Garnsey. She was the project lead from the Pfizer side, while I was the project lead from the Postera side. So Postera was responsible for the discoveries, design, and synthesis of compounds, while Pfizer was handling a lot of the virology, the in vitro and vivo assays, the logistics around kind of PK, tox, those types of things. So it was a great collaboration, and really starting out, our goal was, trying to find new antivirals against currently undrugged viral targets. There was always the possibility of resistance to current antivirals developing over time. So there was great interest in kind of new mechanisms of action. And there was a fair amount of missing knowledge at the project start. So first, could PLpro inhibition result in cellular antiviral activity at therapeutically meaningful potencies? And also kind of related to that, can that inhibition translate to a potent preclinical in vivo efficacy? So can you get the right selectivity and PK properties of these compounds? There was pretty limited literature, chemical matter. GRL 617 was kind of the main literature. This came from an HTS screen on SARS-CoV-1. It's about 1.8 micromolar biochemically in our assay, but had a large drop off to kind of 68 micromolar, even with an efflux inhibitor antivirally. So we knew really a huge jump in potency was needed to show these things. I'll kind of step back just to say, I think, given the diagram I showed at the beginning, a lot of times when I talk about this project, people will say, well, it's obvious, it's essential, it's cutting the polyprotein. But this protein has a really interesting active site where it's the main cysteine that's active is in a very narrow tunnel. It was unclear if covalent inhibitors, like were often used for the main protease, would even be possible. Could you get the potency you needed out of a non-covalent inhibitor? You were competing with not just the polyprotein as a substrate, you were competing with the deubiquinating and ISG-lating activities, which have substrates. So really needed to show this target had the horsepower, as it were, to kill the virus. So to kind of sum up that uncertainty, the existing inhibitors were valuable starting points, but had pretty weak cellular antiviral potencies and minimal information on ADMET. From the work on nirmatrelvir, which I definitely suggest you all read by my collaborators at Pfizer, their paper on this, we had a pretty strong hypothesis that we would need to maintain unbound Cmin greater than EC90, as with other protease inhibitors and MPRO. So this is a plot of kind of the in vivo efficacy from that paper. And the combination of requisite potency and PK was likely going to be difficult, especially in a rodent. Just C-trough indications can be that way sometimes. So given there was really a vast improvement in chemical matter needed, we really tried to leverage our machine learning-based approach to take quite exploratory beginnings to see where we could get large jumps in potency. So namely, we use machine learning to help scan large regions of chemical space, probe multiple synthetically accessible vectors, and diversity select compounds for each library based on bioactivity predictions. So a lot of this is wrapped up in a tool we have internally at Postera called Saber, which provides synthetically accessible bioisostere replacements. So that's really combining the synthetic understanding in an algorithm with bioactivity predictions. So just to show what that interface looks like, this is in the platform we use internally. Essentially, what's beautiful about it is you really just have to draw your parallel medicinal chemistry route, and it will understand all the synthetic requirements. You can then select your building block of interest to replace, and it will essentially use machine learning to suggest bioisosteric building blocks that will help form products with predicted good bioactivity that will meet all the synthetic requirements. So this is really quite nice for designing libraries very quickly with machine learning guided predictions. And it even understands multiple steps and the synthetic requirements that would be involved in deprotections and couplings up multiple steps up the retrosynthetic tree. So just to kind of put this into reality and what this looked like. So an early approach was essentially taking the scaffold here that was started with GRL 617 and building some parallel libraries off this. So this one, for example, is a 50-member library where a Suzuki coupling was done into a new vector. And you'll see a key part of our approach is that we do make quite a bit of inactive compounds. And what we really care about are kind of the tails of the distribution. So are we improving potency at the edges of the distribution? You really only need one really good compound to jump forward in the project. So for example, that was found with a pyrazole here. It gave over a 10-fold jump in biochemical potency. The antiviral potency jumped to about 1.5 micromolar, so still not fantastic, which I'll talk a little bit about, but clearly an improvement, and we maintained quite good ADMET properties. So just kind of to step back and take a look at how we think about these parallel libraries, which are a fair number of compounds and easy synthesis. Kind of the money ball behind this, and Alpha goes into this a lot, is assume you're making n compounds in a batch and you really have a probability P, a compound is a breakthrough. Then what is the probability you get a breakthrough from this large batch of compounds? And you can break that down to this formula if you take the assumption of independence as a simplifying assumption. And what this means is essentially if you crunch it out, the chance of improvement with at least one of 15 compounds with just a 5% chance of working can in fact be higher than a more custom designed, custom synthesis compound with a 50% chance of working. So what this means internally is we spend a lot of time thinking about balancing the number of shots on goal with the quality of the shot we're taking. So you can think of this as if I want to roll a one, should I just roll one standard die or should I roll many like 60-sided die, which you can find in kind of odd board games? So balancing that is part of what we think about a lot. And at Postera, we really believe it's important to understand that the following are imperfect proxy metrics, though they're used quite frequently in medicinal chemistry. So things like number of compounds made, number of iteration cycles, or even the elegance of an experiment, um, often I think are helpful proxies, but maybe in contrast to what really matters, which is the time and cost needed to reach a goal. Obviously, those are often valued differently among organizations or projects, um, but understanding that trade-off is really what we focus on. So a good algorithm can actually be one that suggests the synthesis of many seemingly inelegant molecules as long as you achieve the desired profile in less time as a more seemingly elegant approach with a kind of custom synthesis, as long as resources are equivalent. So to put that into practice, we continued this exploratory approach. Further libraries were enabled. So again, this was a 20-member library with a kind of standard Buchwald coupling, rather simple chemistry to replace the aniline. That provided a pretty big jump in potency biochemically. We are down now to nanomolar antiviral potency. I'll mention these are all plus efflux inhibitor. So we really needed that to see the potency in the high throughput antiviral assay. And then chiromethyl to cyclopropyl switch provided PF 0795742, which was down at 2 nanomolar biochemically. These are KIs since we bottomed out the biochemical assay, and that got to around 150 nanomolar EC50 in the Vero cells. So that was a really nice find, and we were very happy with how quickly we were able to progress that. I do want to show something that at Postera we track very closely, which is just how are things going over time. So here we have both the biochemical potency in blue, and the green potency is the antiviral potency. The line basically sketching out our best compound over time. So you can see within basically 8 months of chemistry, we were able to find that compound, and we were really quite pleased. Now, to make sure we weren't just optimizing potency, we also used this metric derived from the dose score with some simplifying assumptions around the FAFG of the compounds. And we can show those are also vastly improving. So we ended up, I will mention, I'm just presenting one of several series, and this one bottomed out the biochemical assay pretty quickly. We did eventually find other series that kind of got to even picomolar biochemically and close to that antivirally. But it was really nice to see that result where we could take this in vivo. And so just, I really want to give a shout out to a lot of the people who made the logistics on this project flow very smoothly. So at Postera, we really focus on, the models and the ideas, but also how we get information back into those models for improvement. So we think about this as a cycle towards compound improvement, and it should be like a self-sustaining flywheel where it's continually improving with each step improving the next. And so the goal here is, can we really eliminate friction that would basically halt that continual cycle of improvement and design, make, and test. So what that looked like in practice was a lot of work was done in collaboration with the External Research Solutions Group at Pfizer to co-localize assays, reduce cycle times for key antiviral ADMET data. And that really was kind of a jump forward for the project. So in terms of kind of the money ball behind that, in terms of improvement, we think about both the probability that a given cycle of design, make, test will improve on the chemical matter, but also the number of times we can actually compound that cycle over and over again. So a lot of people, I think, focus just on the algorithmic part, perhaps too much, which is generally P, which we also focus a lot on, but I think, it often doesn't get enough focus how much improving your number of times you can compound and your cycle time also helps with that progress. So in terms of the profile of PFO 79742, which was found, we're very pleased to see this was potent, selective, and orally available across species. So besides the Vero assay, this also went into a human airway epithelial primary cell assay, which is shown up here in the top left. We saw an improvement as we went to the primary cells, and this was eventually kind of the dose setting assay for the in vivo experiment. So that's 13 nanomolar EC50 and around a 60 nanomolar EC90. In terms of off-target pharmacology, this was quite clean in a SERP off-target panel that was quite broad. Protease panel, it was very clean. Ion transporters, pretty clean. hERG, we had a 28 micromolar signal, which was judged to be have sufficient margins due to the compounds being quite potent. Looked pretty decent across CYPs, very soluble. There is some efflux in MDR1, which is why early on we needed the efflux inhibitor in the Vero cells to really shut down the PGP and see that early potency. But we were able to maintain pretty good absorption across the board in multiple species, moderate to high volumes, and then pretty low turnover. So in terms of what we can show here, I will mention in the spirit of kind of the open science antivirals, some experience we found was that the biochemical to cell drop-off was quite consistent across the board. So kind of 100-fold drop-off. So as you can see, the Vero EC50 is around 147 nanomolar. And then when we went to primary cells, that really dropped down to 14 nanomolar. So we track this quite closely, and generally across compounds, you see this biochemical to cell drop-off in the Vero assay about 100x. I'll mention around this part of the project, when you're reaching 10 nanomolar biochemically and still seeing 1 to 10 micromolar in an antiviral assay, that can be quite nerve-wracking, because those are also the potencies you often see off-target effects give. There's a lot of discussion about that and, what could be the issue of the drop-off. Eventually, it was basically solved by just making the compounds even more potent and then things straightened out. But clearly a lot of uncertainty with viral projects when in that space, and I've seen that on multiple programs. So luckily, this compound had pretty good rodent PK in order to go into the mouse model. We thought we had good coverage over the primary cell, EC90 in the HAE cells, and we aimed for oral BID doses, giving intended Cmin, unbound Cmin of 1/3 x, 1x, and 3x the EC90. So we wanted a subtherapeutic and supratherapeutic dose so we could really see a dose response. I think that's something that perhaps isn't done enough in the literature, and I really encourage it because it gives a way to correlate kind of the potency you see in vitro with the in vivo lung viral titer reduction. So luckily, as you can see here, the compound was quite efficacious in this mouse model of infection. The mice were infected 4 hours prior to the first dose, dosed BID for 4 days, and then the lung viral titers were measured on day 4. The control here is nirmatrelvir at 1,000 mg per kg, which was used previously in the paper on nirmatrelvir. So a really nice thing to see is also that the infected and untreated mice had robust infection, and that was also similar to the levels in the subtherapeutic dose. So again, I think it's really important to see that you have similar levels of robust infection across the board as a control, and that you really see this dose response. So we were very happy with this result. And then we even saw pretty good correlation of compound potency from the in vitro HAE assay to the in vivo lung titer reduction. So you can draw a dose response from the in vivo data and see that the modeled EC90s are almost exactly what we saw in the primary cells. So that was really great to see from modeling perspective. In terms of immunohistochemistry and lung histopath, this also looked pretty good with the highest doses giving relatively clean images. And we also saw good protection against the weight loss that you get in infected mice. So all this data lined up very nicely. So just to kind of give a summary of this compound that we're releasing as a tool compound, I think it's a really valuable in vitro and in vivo tool for further study of PLpro. It has favorable PK and an in vitro profile that allows us to show PLpro is essential and druggable as a target. Importantly, this is a differentiated mechanism to existing antivirals and crucially still possesses robust single agent efficacy in this in vivo model of infection. So that, again, was really nice to see. There are some open questions which hopefully can be answered, with this valuable compound being out there. So could a combination approach potentially offer additional benefit in a clinical setting? I think is a really nice question, along with the degree of benefit that this immunomodulatory effect can confer in an in vivo setting, which I'm not sure is exactly clear yet. So thanks, really appreciate everyone taking the time to listen. Thanks to the entire Postera team, many of whom were on this project, and to all who are involved in this project across the collaboration. So Pfizer, WuXi, and Utah State University. So a lot of people to put this nice data package together. Thanks.